Assalamu alaikum. This is an introduction to immunology by me, Dr. Sarah Abu Zaid, PhD in immunology from the Meta Faculty of Science, Egypt. History of immunology. With the evolution of the germ theory of disease, the study of the mechanisms of immunity and the possible consequence of infectious diseases began almost simultaneously. Interest in immunology grew out of the everyday evidence that those individuals who survived bogged and disfigured from the dread disease smallpox never did contract the infectious again. As early as 1773, Voltaire reported on an ancient Chinese custom where dried and powdered smallpox scabs were inhaled. Although it would take several years before bacteria and viruses were fully characterized, theorists were already elucidating the mechanisms of host defense against infectious diseases and their possible prevention. The phagocytic theory was the first to be developed in the 1880, a bold and imaginative concept, the fruit of Eli Mechnikov, deep knowledge of biology. Mechnikov hypothesized that the basis of inflammation was the cellular reaction and that vascular and nervous reactions were only of secondary importance. He postulated further that these migrating cells, which were able to move in order to meet an enemy, were the major guardians of health against bacterial infections. Louis Pasteur reported on the treatment of his first two patients with rabies using early vaccines in 1888. Mechnikov left Russia to continue his work on phagocytosis in Pasteur's Institute in Paris. In 1908, he shared the Nobel Prize with Paul Ehrlich for his early contributions to the understanding of inflammation. In 1903, almost right with his disciple Stewart Douglas, observed that a humoral component which they designated opsonin called render bacteria susceptible to phagocytosis. They thus forged a link between the two major theories of immunity. Since that time, humoral and cellular components were shown to be interwoven in an intricate pattern. The complex chemicals associated with immunity have been the continuing subject of years of research. Emil von Behring and Kitasato in 1890 inoculated animals with toxins of diphtheria and tetanus to produce neutralizing antitoxin serum. They introduced passive immunization into modern medicine, and for this, von Behring was awarded the French Legion of Honor and the Nobel Prize in 1901. Beefeater in 1894 and 95 discovered the phenomenon of in vivo cytolysis of Vibrio cholera when the organism and the immune serum interacted intraperitoneally in the guinea pig. From these early experiments, the nature and function of complement-mediated cytolysis were elucidated by Favre and later by Beckner and Bordet. Jules Bordet was awarded the Nobel Prize in 1920 for his pioneering work on complement, for his discovery of the whooping cough agent, and for his enunciation of the diverse aspects, antigen-antibody reactions, and the blood coagulation system. Fernand Widel, the Allergian-born son of French army surgeon, described the agglutination reaction for typhoid fever, a test which is still widely used and bears his name. In the early 19th century, the practice of transfusion of blood from man to man remained a risky, unpredictable business when at the turn of the century Karl Landsteiner discovered the blood groups. Landsteiner received the Nobel Prize in 1930 for elucidating the blood groups and for his work on the Rh factor. As the serotherapy developed, it soon became evident that antigen-antibody reactions in vivo could have some harmful effects and even produce death. Anaphylaxis, the most dramatic manifestation of hypersensitivity, was first described by Portier and Carles Richard in 1902. In 1913, Carles Richard received the Nobel Prize in recognition of his work on anaphylaxis. The 20th century saw phenomenal progress in the understanding of immunological concepts. How antigen direct the various immunological processes was the chief point of contention among early theorists who had to choose between the instructive and the selective theories. In the past 30 years, the biological basis of the immune response was in part clarified when the theory of clonal selection of antibody formation found acceptance. This theory, proposed in essence by Paul Ehrlich almost 100 years ago, fell out of favor with the introduction of the template theory. The first comprehensive attack against the template theory was launched in 1955 by Niels Jern, who recalled certain criticisms made by Macfarlane Burnett against the template theory. It, however, remained for Burnett to draw together the new conceptualization, and in 1957 he asserted that each cell and its clones can produce just one kind of receptor to explain the specificity of the immune response and the exponential rise in antibody production following contact with antigen. The secondary response is more powerful because antigenic memory leads to rapid clonal expansion during subsequent contact with the same antigen. Macfarlane Burnett was awarded the Nobel Prize in 1960, which he shared with Peter Medawar. In 1984, Niels Jern was also awarded the Nobel Prize for his theoretical contributions to immunology, the most fundamental of which was his role in developing the concept of coloniality and for his description of the idiotype network in the regulation of immune responses. The elucidation of antibody structure is credited to Rodney Porter and Gerald Edelman, who shared the Nobel Prize for their discovery in 1972. The production of monoclonal antibodies by somatic cell hybridization of antibody-forming cells and continuously replicating cell lines was called the technique of hybridoma formation. This technique, described by George Kohler and Cesar Milstein in 1975, he enabled immunologists to prepare virtually unlimited quantities of antibodies that are chemically, physically, and immunologically completely homogeneous. For this invaluable contribution to immunology, they were awarded the Nobel Prize in 1984. Existence of markers of biological individuality, what we know as histocompatibility antigens, was first suggested by Gorer in 1937, who demonstrated that loci determining blood groups antigens and those controlling tumor rejection in mice were distinct. Snell in 1948 he showed that there were multiple alleles in the mouse H2 locus, and subsequently that this locus was genetically complex. The H2 locus was shown to control the phenotype expression of certain cell surface markers in mice. In 1950, Dosset identified the HLA human leukocyte antigen locus, or MHC, major histocompatibility complex as it is sometimes called. Benicraft showed that genes of the HLA determining loci may control immune responses. Neil Dosset and Pencroff were awarded the Nobel Prize in 1980. Peter Doherty with Rolf Zinckernagel found that T cells from mice infected with the meningitis virus destroyed virus infected cells only from the same strain of mice, and they showed that T cells must recognize two signals on an infected cell, one from the virus and one from the cell's own antigens to destroy it. For this new understanding of cellular immune mechanisms, they shared the Nobel Prize in 1996. Immunology continues to fascinate and frustrate researchers the world over. A better understanding of tumor immunology, autoimmunity, and response to immunization could develop into strategies for protection against many crippling diseases. Thank you.